got a move going?
We're, yep, 9 meters left. I'll put in another.
So related to the Niskin bottle, so we're collecting water samples, and each of those that we hope will tell us quite a wide variety of things. One of them, something called eDNA, where we sample through the water and analyze it for DNA. So all organisms, as they're traveling through the water's passing through them. In the case of the sponges and the tunicates and the corals that we see, slough off little bits of their cells and DNA. And if we can capture that, then that can tell us what all's been in the water. So these surveys that we're seeing right now are incredibly important and valuable in terms of just what we see on the video and what you're seeing right now. But we're missing a ton. We don't know what's just 2 meters on either side of the vehicle. We can only see just what's sort of in front of us and what you're seeing on the screen now. But the DNA will tell us all the things that are in that area, ranging from small bacteria or plankton to these large organisms and fish. So that's one thing we're interested in. Another thing is we can analyze those samples for microplastics. So all the plastic material that ends up in the ocean one way or another.
I'll just do that. All right. Thanks,
breaks down into tinier and tinier and tinier particles. And some creams and things actually have little bits of plastic intentionally in them, scrubs for your face. And those particles can make it into the water from your shower and your drain all the way into the ocean. A lot of them are now banned and outlawed. But they make it in the ocean and they make it through the food web, make it into the water. And so we're getting a feeling or sense for at what level we see those microplastics here in the Channel Islands. And some studies have already been done up and down our coast that show pretty much everywhere you go, if you take a sample of water, you'll find little bits of plastic in it.
So sure, I'll just jump in here for a second and say we have come down about 20 meters, and it does seem like the environment has totally changed here.
Mm-hmm.
Yeah, this is great, and the backscatter's definitely been fantastic for this. So I guess it looks like we'll be rising up again. If we could pass through the EF2B marker and head, looking at HYPACK, head to about where the B is, and then we can make a decision on where to go from there. Does that sound all right?
That sounds great. Yeah, it looks like we're starting to head up slope again now from our sonar, also from the video.
Little flat fish in the screen, maybe 25 centimeters or so. So those lasers, for those that are looking, are 10 centimeters apart, the two green dots in the middle of your screen. We use them to measure the animals that we see, get a sense for how big they are. In some cases, we have a good understanding of the relationship between size and age. So if we know how long somebody is, we can also tell roughly how old they are. We know that a lot better for fish than sponges or corals, but people are starting to figure that out as well.
Thank you. I think the last 20 questions here are all people who want to come out and work on the ship or be a scientist and do the work that you and Lizzie are doing. So yeah. lots of comments about, I want to do this when I grow up.
We need all the help we can get.
Good. There's a question about if what we're seeing with the glass sponges could be anything analogous to coral bleaching, like what we're. I don't know if that's a question you can answer. If Abby might be able to respond to that one.
Yeah, we can throw that one back out to Abby, and I can comment just a little bit on the shallow water coral side of things. Coral bleaching occurs when the coral gets stressed, usually because of an increase of temperature that occurs for a certain period of time, a number of days. And if they're exposed to water temperatures above that threshold for enough days, when they get stressed, they release their symbiotic zooxanthellae, a dinoflagellate, a little micro organism of plankton that lives within their tissue and helps helps them feed. It becomes part of their food source. And so in this case, these organisms aren't living symbiotically with an algae in the same way. So we're trying to figure out other reasons that they might be stressed, but one of them very similarly, it's an excellent question, might be, we heard from Abby, our sponge expert earlier, temperature. So the temperature of the ocean we know has been increasing, certainly in shallow waters, resulting in the coral bleaching that you hear about and have seen pictures of, called bleaching because when they lose the algae is what gives them their color, and so they turn very white. They look like their skeleton underneath. And in these deeper waters, similarly, an increase in temperature could cause a stress that might cause these sponges to die. So that's certainly a factor that would be worth looking into. Others she's hypothesized included, I'm trying to think back of her text, sedimentation was one. So if we had a big sediment event that covered them over, that might have affected them, as well as changing oxygen levels. So if the oxygen in the environment changes for any reason over a period of time, these animals can't move to get to other places where there's less sediment or better temperature or more oxygen, and so that could have resulted in the mortality that we see. But we don't know. So excellent question, and similar in a way.
Thanks. That was a great answer. This looks like we're getting into another interesting area here with more.
Wow. Yeah.
More sponges.
Very large sponges.
They are stunning.
This is all so cool. You're all at home seeing this with us for the very first time. You are discovering this as we discover this, which is awesome.
There are so many people who are just steadfast viewers, people who are saying this is so much better than just watching YouTube videos. So
Is
like
Saturday Night Live on yet? What are we competing against?
Yeah. Yeah. Funny. Let's see. There's a question about do the lights of the lasers disturb the animals, or lights from the Hercules disturb the animals and result in changing their behavior in any way that you're aware of?
Yeah, the ROV definitely makes some noise. There are thrusters, so it's traveling and propelling itself through the water. So not only is there sound, but there's also mechanical vibrations in the water that fish and other organisms can sense that might alert them or tip them off that it's time to run away. And then, as you mentioned, as this watcher mentioned as well, the lights could also be quite uncomfortable or surprising for some organisms here. And as you can see, there's this rockfish here on the left side of the screen who doesn't seem particularly perturbed by our presence. And I'd say that's been true for several rockfish species that we've encountered. And some of the species that run away from the ROV will never know what they are because they see us and they hear us coming before they're ever in view of the Hercules camera. So there are ways to kind of test that. You could use a drop camera, which is a stationary camera you leave on the bottom of the seafloor that takes pictures at different intervals and has different light sources, like red light instead of light on the visible spectrum like we're seeing now, or infrared light is another option. But with the ROV, there are some certain limitations and drawbacks, with just being a big, noisy ROV coming through the water column. But we do still get a lot of information out of these visual surveys. So it's just something we have to take into consideration. Do you have anything to add, Chris or Nautilus? Peeps?
I would say it's always hard to characterize what's there when you're not watching, right? It's easy to say what's there when you are watching, but the reverse is, it's always hard.
Mm-hmm. Yeah, and for the stationary cameras, which is kind of a way to get at that question a little bit, that is stationary. So you're only getting a small snapshot of what happens to be in that area when the camera is taking a picture. So there's no one way to collect data to rule them all to answer who's there and why.
The ROVs travel through space, and the drop cams stay in one place, but you get the nice time series. But there's no good way to do both, maybe.
So shoreside, we're about 10 meters off from completing this move, which will take us about 40 meters east of EF2B. What are you thinking for our next move?
I mean, we could keep going east, Chris, if you wanted to. We could go see if there's any more dead sponge, because essentially this is the sponge reef that I was referring to, that I wanted to get to eventually when the ROV first hit bottom for the previous dive. So I am happy as a clam right now. We're already at the target that I wanted. So I think just more exploring for me would make me happy, but are there any specific directions that you would want to hit or go to for the remainder of our dive?
Happy as a shingle shell clam, in fact.
A shingle shell?
I learned that term from Daniel yesterday. So question, did we want to, are we happy with the small mosaic we got there, or do we want a landscape mosaic over some of this reef? Because this is as good an area as any to do it if we did, is one question. And number two, I think we want to prioritize, but it definitely does not have to happen on our shift, but we would need to provide some direction to the next crew coming in, that we'd want them to go back to this glass sponge reef, and get us something there. So yeah, two quick questions.
Yeah, so the habitat mosaic, we did a 10 by 10 meter last night, Lisa and Di did, and originally we started out 15 by 15, and that was going to take 4 hours, and the 10 by 10 took 3 hours. So I'm not sure if that is how we want to spend our time here.
Yeah, I know.
So I think we did capture a lot of these same species that we did on the Boulder 3D that we just did. So I think I'm happy with that.
Okay. So then one of my thoughts would be to characterize this place. We could do, this is pretty amazing. It would be great to have
we should start a
quantitative
transect. Yeah.
So we should do a transect to give them a heading, and then maybe tell the next shift, it's up to them to go back to the, so maybe we can explore. I'm just going to look at the rest of your waypoints here.
Yeah, so if you look at HYPACK or my map, if we're at EF2B or slightly east of it, if we go pretty much straight south, that's towards waypoint U, which is where we initially landed in this sponge garden. So if we want to do a 200-meter transect in that direction, I'm pretty sure we'll have a good shot of staying within this same habitat, if that's where you want to put your transect, and then we can decide where to go from there. Or if we don't want to get too far away from the glass sponge reefs, because that would mean they'd have to transit back in that direction, maybe we can go west. I don't know. That's a good question. Or continue east? We could just continue east too.
The only other thing I'll add in here is I'm flipping on the slope map for on HYPACK, and it does look like we're about to go down slope again. So I think we probably want to stay, you know, if you're looking to continue along this reef, I think we do want to head south, but almost a southwest slight angle to get
Yeah.
down there so we don't lose the slope, or we don't lose this ridge.
Yeah.
Mm-hmm.
Yeah. I like that idea, Lizzie. What if
Yeah.
south being more optimal, because either we might come back into contact with the Ferrea Reef, or from there versus going east, or from there we won't be just further away from that reef. They can just book it, which should be an interesting transect, straight back to it. Yeah, we could do kind of a, we could go south for 200 and then south for 200, west for 200, and north for 200, and that'll put them pretty close back to the Ferrea Reef and explore this pretty large area of backscatter. So why don't we do the, that heading that you're showing there, somewhere around that looks fantastic. We can reconsider when we get to the end, but a quantitative transect will be great.
Okay, and did you want to do a due south or more of a southwest?
That slight southwest looked pretty appealing to me.
Let me do it again, just in case we had a delay. I had it kind of in between EFG2, or?
Yeah.
Okay, perfect.
I like that. Yeah, perfect.
And then when you mentioned doing several transects, would those be back-to-back, or would we keep 100 meters in between?
Sorry, we'll decide, I guess, if we want to do another transect right away. We would definitely want to space that out. So we'll do this 200 transect and then probably do a little bit more exploration before we hit another one.
Okay, perfect. Thanks.
And Lizzie, one of the things I'm thinking about that they could do on the Ferrea Reef is a transect. I think it'd be great to have a transect on that reef and a mosaic, so we can give them two tasks. So we'll get them close. We'll do this transect. We'll have time to get them a little bit closer, and then they'll have two tasks to kind of wrap things up in addition to the exploration that they want to do. Does that sound good?
Yeah. We have a request for a Zoom before we start a transect, so I'm trying to see if we're still in that area.
Sure thing. Do you want to grab a Zoom right here on anything?
She's looking for a plate sponge. And she was saying
sponge
right off the bottom of the screen. But I guess I'm not sure how much we moved between the last time she sent that message. So she said just bump it back a little bit.
Back a little bit. Got it. And Paul's on Hercules right now, so you know shore side.
Thank you. Hey, Paul.
How's it going?
Pretty darn good. Sponge City up in here.
Amazing, yeah.
So we think the sponge is just kind of below us? Behind us?
Yeah, just below us. It's a plate-looking sponge. Maybe that one to the left? Is that platey, or just the one below us?
Erica says yes.
Yeah, so maybe pause here and we can zoom in on what's right in front of us.
It's this one right in front of us, or the...
Yeah, they're just below the lasers. There's a barrel, and then to the right of that, down and to the right, it looks like a plate.
Copy that.
Kind of looks like an upside-down salad bowl, but maybe
yeah,
it's flatter than it looks.
And those do tend to do that.
My sponge identification is mostly in terms of either salad bowls or dirtiness levels.
Yeah, dishware or...
Yeah.
This is a great...
That's all legit.
Yeah. Hey, Laura, before you go, are you still with us? Do we want to end on one final question so we don't take up too much of your time?
Sure. Yeah. And I'll be back again at 9 a.m. tomorrow, too, for everyone out there that if we didn't get to your question, we'll try to get to it then. So, yeah, there's a quick question here about, are the tubes that you see in the sponges, are they designed to catch food, or do they just happen to grow that way?
Oh, so sponges have many different morphologies, and the tubes are meant to catch
let's
food.
zoom in.
They have a bunch of little pores. That's why their phylum is called Porifera. They have a bunch of tiny little pores that draw water within those larger tubes, and then that come out the sides. And so sponges can be one big tube, and we call that a barrel, or they can have many little tubes. They've just evolved to have different body plans like that, which may confer some kind of benefit to have multiple tubes, but I couldn't say as a non-sponge expert.
And is this what we were
Thank you
looking
very much.
for with this zoom?
I believe so.
And were there any samples that we needed around here, or just the zoom?
Waiting to see if they in the science chat. Yes, can we sample this sponge, please?
Okay, let's come right out.
All right, Lizzie, I'm going to sign off, but I'll be back. I think you're on shift again tomorrow. You're on
Are we
watch at
close
9
enough, or
a.m.,
do
so
we
I'll
want to
chat
bump
with you then.
forward, Bob, to sample that sponge on
That
the
sounds
lasers?
wonderful. I look forward to it. Thank you, Laura, for answering or for helping us answer some of those public questions. That was great.
Great job,
Thank
Laura.
you.
Thanks. That was fun.
Yeah, thank all of you. Bye-bye. Enjoy your evening.
Good night.
So, yeah. Do we know what kind of sample this sponge is?
Is this a grab and slurp, Abby and Erica? So it's saying grab, snip a piece if possible.
Okay.
Crab on the front
Abby
porch.
saying if we could from the bottom or the left side to take a snip, that would be great. So part of the sponge looks a little bit degraded on the upper right, and so I think to identify the spicules, she would like a piece from the bottom or the left, which looks a bit more intact. And she's saying because it looks degraded, there's a chance that it's actually dying already and may not be attached to the seafloor. Steve, if this whole sponge does come up because it's already on its way out, would this be one of those samples that would potentially be difficult to dry before the end of the cruise?
You know, I've had mixed results so far. The sponges from this morning, I put actually right in front of the flow of the air conditioner in the wet lab and actually dried them out in about 12 hours, but I would still want to give it, 24 solid hours to 2 days, which we could do. So it is possible to still dry it depending on how big and wet it is.
Okay.
But we'll take
So,
the voucher for DNA as well.
Okay.
I don't know which corner we're going to be able to cut off with these snips, do you know? There's no easy part sticking out.
Oh, he's trying to hold the manipulator hand.
I don't know about snipping. Maybe you just
Kind of grab it.
grab and you probably got to turn the grip force down. But I don't think it's going to close less than 5. So
Go 6.
Yeah, just try it and see how slow it is.
Oh my gosh, you have these crabs that are after that manipulator arm.
Let me test. Actually, can you come wide? I'm just going to test the claw here. So that's on 6.
Yeah, so maybe go to a 5. I don't know.
I think what's happening is that these crabs, they perch on top of the colonies like this, or the sponges or corals, and they're really trying to capture midwater organisms like fish as they drift down, and so it's seeing a shade,
Oh, really?
and it's going to try and grab whatever's in front of it, thinking it's a meal.
He's like, whoa, that's not a fish.
We've actually seen this a lot on some of the Central Pacific seamounts where, you know, a shadow goes by, for example, and all the crabs start closing their claws and reaching out. It's really funny.
Oh my gosh.
So that was 5. That might have been too slow.
Yeah.
All right, I'm going to go for 6 and just try to grab this section of sponge then. Can we zoom in a little bit, Al?
Oh my gosh. You're barking up the wrong tree, buddy.
Oh yeah, the whole thing seems pretty loose.
Yeah, it even might be an upside down salad bowl sponge like we had been seeing.
We have a big box if you want to get it.
Yeah, I think so. If it's already detached and dying, I think Abby would be happy to have it. Yeah, it's broken.
We're going to go on the starboard side with this in box E.
Okay, let me pull
Don't
up.
really have a grab on it yet.
Nope.
Don't do it.
Oh my gosh.
You might get a bonus crab.
Oh no. Don't get attached, crab.
Sometimes you just go like. There was a crab just hanging out on the front porch as well. Just kind of sitting there. Along for the ride. This looks a lot more flimsy than the salad bowl. The salad bowl had some rigidity to it.
Okay. Perhaps it is a plate then.
I only know that because we handled it in the lab this morning, so I'm not sure if anyone's seen the salad bowl before.
Do we want to zoom on this before we take it in?
Sure.
This crab is very curious about the size of that claw. How does it work?
He just doesn't know what to do.
Oh, almost got rid of him.
Yeah, I'm not sure if the whole thing is going to make it to the journey over there.
Oh, good. See you later.
It's because we're stuck with this higher grip force, kind of just wants to tear off, so we might only end up with this chunk.
Either way, we have to go to the starboard box. We have sponges in the forward anyway.
Copy that. Can we zoom out here, Al?
Can you zoom in on Argus? You could go in more. Just to give him a side view there.
Is that, oh yeah, I'm clear of the light bar.
Ready for sample mode?
Yeah. Can we switch the second camera too? Yeah, it's just hanging on by a
Barely.
thread.
Yeah. You gotta go up where I open the drawer.
Oh.
There it goes. Is that enough of a piece?
I think that's enough of a piece, yeah.
Okay, I need to turn off the thruster there too. So, you frozen?
Yep, I'm frozen.
Well, so you
Oh.
get ready to drive down more because I'm going to take off your thruster.
Do I have to take off stick lock to drive down more?
Yeah.
Right, yeah.
Or you could just up your joy gain. Sorry. Just up the joy gain. You don't have to monkey with nothing.
Okay.
Are you ready?
Yep.
It's off. Might be might settle out okay. Yeah. Okay. You ready
All right.
for drawer?
Yeah, I'm up above it, right?
Yeah.
So?
Yeah.
Coming out.
Which one's it going in?
We can go in the outer compartment E.
Okay.
Looks good.
Yeah, just trying to close the jaw again.
Okay. Closing. You can watch in the Argus. Yeah. Ready to switch back?
Yeah.
And you come down with the elbow. See, that's how you hit the corner. Is if you have the elbow and the shoulder up,
Mm.
and you're reaching too high in the sky.
Is that good?
Yep.
For stowing it?
That'll do.
Okay.
Okay,
And is that
turn
a
that on, Argus?
thruster back on too?
Yep. Ready? There.
All right, let me just get squared up here.
Shoreside, anything else we want to zoom in or sample on here, or are you ready to start the transect?
As far as I know, I think we're good.
Alrighty.
Okay. Let me just swing around and get in
away.
front before we call on that ship move.
Sounds good.
What'd you say the bearing was going to be?
186. We can do 185 if that's easier.
was at 100. I knew it felt a little bit zippy.
Quick pirouette.
All right, Bob, I'm going to kind of come around and
All right.
underneath you.
So pilots, this transect will take us upslope. Nothing too steep, but probably more or less what we're looking at right now. Ready for a ship move?
Yeah.
Bridge, nav. Step 200 meters, bearing 185. Can you repeat data?
Can you check back? Got it.
Ship move is underway. Let me know
Okay.
when you start moving forward and I'll drop a target. Steve, did I just say transect 8 to you?
Yeah.
Thanks.
Yeah, copy that. Transect 8. That's what I've got too.
Perfect. This is an interesting formation here.
Okay, I think we're
forward.
moving forward now.
On the way, transect 8. Getting that noise in the Argus sonar again. Haven't seen that in a while.
I think it's interference from other sound emitting instruments.
Ah.
So it gets in sync with those other things at the wrong time. You know?
Are those other things from us or from other?
They're from us.
Okay.
Yeah. So, yeah.
Interesting. We're definitely moving up slope as promised.
So what we really need is
Yeah, this looks great.
we need to be able to trigger all the sound emitting devices, be able to control when they transmit. So then if you make them all transmit at the same time, there's a blanking interval.
Yeah.
So,
Oh, cool.
yeah. Then they don't step on each other.
Yeah.
Get that in a Herc too.
Yeah? Believe it when I see it.
It's pretty easy to do, really.
Oh yeah, no, I believe you on that. It was
Yeah.
the Herc too.
Oh, the Herc 2, yeah. I mean, we could do it in Herc 1, but
Yeah.
I think there's not a lot of interest in, in working on Herc 1.
The current just pick up a little bit?
It's been higher than it was
Yeah.
like an hour ago.
I guess we're also now moving directly into it, where before it was coming from the east.
No, it was blowing to the east, right? I mean, that's the way it's been.
Yeah.
Yeah. So we're going crosswise to it now.
Huh. Okay. I guess we're moving forward, so the particles are coming straight at us.
I mean, is the currents here, are they tidal?
I don't think so.
No? It's just constantly blowing the... I mean, it was very consistent on that, on our last watch, 090.
Yeah. We're moving south. But yes, it was super constant east.
Yeah.
My dashboard on that doesn't work. Do you guys actually have a measure for what the current is right now?
We do not. We know the surface current.
Yeah, surface current 0.7 knots coming from 286. So.
Okay.
Heading east, but we don't have any sensors to determine current below, as Bob's been joking. Best way to do it is let go of the controls for a minute on Herc and see how quickly we drift. Which did work earlier this morning or last night, some point.
Yeah.
Maybe we'll do that after the transect.
Yeah. Yeah, not a great time.
Well, we go hands off a lot, though.
Yeah.
I mean, on most of this, the last few watches, we've just, like if we're going east, we just let go of the horizontal thrusters and just move with the current.
Yeah.
Okay.
What looks like Bob just driving extremely straight is auto headings.
Yeah. We also have autopilot, so.
Yeah.
Which occasionally will malfunction because of, it loses track of the bottom, and that's when you go into crash mode.
It's not a good mode.
Okay.
It's not as sophisticated as a 737 MAX.
Oh.
The technology is pretty amazing.
We have comments in the science chat. Well, I'm really glad I'm not the one that has to count this transect.
I mean, what's our guesstimate of how many different species we've seen just in this reef? Like
1, 2, 3, 4, 5, 6, 7, 8.
1,000,001, 1,000,002.
Yeah.
Exactly. It's really, looking at it, it's like something out of Alice in Wonderland. I mean, some of these are just absolutely crazy looks to them and morphologies to them.
Too. Oh, we see a lot of small sponges, but like these giant plate sponges are amazing.
So Lizzie, you had mentioned that there was a sponge reef that you were really excited to ground truth north of us. How did that come about? How did you identify that as a site that had a sponge reef, or that could potentially be a sponge reef?
That was where we landed at the 4 a.m. shift when we started the footprint dive.
Ah, okay. Yep.
I knew it was a sponge reef because Tom had reported out, and then I rewound in the Nautilus Live, and it pretty much looked just like this. So I feel like it must be an extension of that, which means it's absolutely massive because we landed at point U, which I think you can probably see. You have dropped targets from yesterday's dive, 1836. So, however many meters we are far from those initial targets is this extends, I'm sure, that whole way.
Got it. Okay.
Maybe patchy, but yeah.
Yeah. Well, I guess we're going to cross over it if it is a big reef moving diagonally. We're going to find out. Very cool.
Yeah. Otherwise, I had no idea, and I just wanted Tom Leidig and I had talked on the phone a few weeks ago when we were initially planning some of these Nautilus dives, and this is my first time being a chief scientist for Nautilus for some portion of this leg, and I was asking him, I was like, well, I have all of these maps. These are on my data layers. Using a combination of these, how do you choose interesting sites? And part of it comes down to where have we not been yet? And given our resources for this cruise, which is, Hercules and Argus, which are extremely capable ROVs to really deep depths. And previous surveys in the sanctuary have been limited to roughly 350 meters, or at least a lot of the surveys have been in the past. And so, wow, look at that massive dead heterocone sponge there on the left. That's incredible. So Footprint Reef has been very well surveyed, and as we found out, is just littered with fishing line, unfortunately. I'm not so sure about Picky Bank because it's further offshore, but that's also a really well characterized site, and the depth in between them was too much for some of the Southwest Fisheries Science Center's submersibles. They're limited to 350, and so he was just saying, it might be interesting to see what's in between. And Nicole had sent me the backscatter from this area that OET has collected over time. And from there, we saw that in between in this valley, there was a lot of hard habitat. And I was just like, well, let's pick some points where we can start deep, head up slope, and end up on top of Footprint. And we just landed smack dab in the middle of an area that looked just like this. So, it's just an educated guess, really, is what it comes down to.
Always exciting when the guess is ground truthed and something very exciting, like this nonstop sponge
definitely.
reef. So that large heterocone sponge, we were talking a little bit with Tom when the watch change was happening, and there was some thought that, the large dead components of the reef were probably something that kind of happened over time. Like there was never, that whole reef was never just alive with living sponges. But what about those big heterocone sponges? That look like one organism, or do those actually kind of grow in layers as well? Or is Abby still around?
Yeah, Abby's chatting in the science chat. We'll see if we get something from her, but they can grow that large as one single organism. Yeah. I think, like this sponge example on the left, it's not quite as big as that heterocone, and there's a little cat shark at the bottom on the left too. I think they can get that large, but I've also seen heterocones that were like similar in size, but they were dying from the base up. Mm. And whether or not they were still growing as their base was dying, I'm not sure.
Yeah, interesting. Like, are they building reefs on themselves?
Sponges.
Super interesting.
Right. Kind of like Lophelia. They're just growing on.
Yeah.
Producing more polyps on top of their dead skeletons.
Can you zoom out on that?
Abby's saying that the modern sponge reefs that we know of are built by the heterocone calyx.
Okay.
Afrocolistes vastus and Phorea.
Zoom back in.
And here we only seem to see them as like singletons quite often, the heterocones. But I do think we saw some amongst that larger reef before. But I think Abby was saying it was mostly the Phorea glass sponge.
So we have about 80 meters left of this transect.
Thank you. Roger that. Abby's following up. She's also not sure what the maximum size or the lifespan of a heterocone is, but from what she read, that the really big ones can be centuries old.
Wow. Very cool.
And yeah, it blows my mind to think that sponges are down here growing at hundreds of years old, not caring about us or anything that we're doing. They're just down here doing their sponge thing, you know?
Yep.
It's a nice double barrel sponge. Almost looks like PVC pipe or something. Very white and clean and crisp. Still not a lot of fish.
The current
We've
shifted
seen
just a
some every
tiny
now and
bit.
then.
Instead of pushing me like that, now it's pushing me like that.
Got it.
Some of that club sponge or the stalked Mycale that just went by. Another massive sponge just
Wow.
there onto the right.
$50 meters left of the transect. So what's next when we complete this transect shoreside?
Ugh, that is a wonderful question. I think Chris had wanted to kind of do a wide loop back towards the dead sponge reef, just so we set the next shift up to do a mosaic. But I think we still have a little bit of time before we need to head back necessarily. So I think after this transect is over, we definitely have a Zoom request.
Alrighty.
And so while we're Zooming and humming and hawing about what to collect, hopefully Chris and I can figure out where to go next.
Sounds great.
I just want to start in the center and then do like a spiral out to everything.
Wow, look at that sponge on the left.
Wow, that's a big one. So, Lizzie, the one thing I'll mention on HYPACK that I did see is that backscatter does show this, what looks like a valley kind of south on the way to EFG too, but it looks kind of funky there. And then when we switch into the bathy, it disappears, and in slope map, it also doesn't suggest that there's actually anything going on there. So I'm kind of thinking that might be an artifact. It's just a weird contour there.
Shape, yeah, definitely.
And the fact it's not showing up on either other map suggests that it might not actually be real. So
Okay. Yeah. We'll keep that in mind. Maybe Chris will want to go into a slightly different direction.
sounds good. Yeah, the slope map, it
I'll wait for him to
Maybe
speak
goes
up.
a little upslope, but doesn't look like there's quite as dramatic a feature as it looked like on the backscatter.
Can you zoom out on HYPACK a little bit so we have a little bit of a
Sure.
where we've come?
Do you want slope,
where we might wanna
bathy, or
go?
backscatter?
I like backscatter. It's proven to be quite accurate
There you
so far. Thank you. Part of me just wants to run upslope towards Piggy Bank, but I'll wait to see what Chris says.
And which feature is Piggy Bank?
Piggy Bank is the one that's directly southwest of us. So you'll see those contour lines that start lighting up that make like a semicircle almost after this gray valley we're about to run into.
Yeah.
So if you were to keep going south according to this map, Piggy Bank is just a big dome that it's covered in hard bottom, and there's been lots of surveys there with AUVs and ROVs, and there's lots of corals and sponges.
Got it.
How much is left on this transect, Sam?
17 meters.
Got it. Thank you.
So Lizzie, after this, do some zooms, pull a Niskin bottle, and what do you think about heading to EFG2?
Yeah, we can keep on that course. I know that's what we originally discussed.
That would cross us through the darker portion of the backscatter, which would be of interest. And then we could kind of see what that chunk of reef looks like over there. And then that would be relatively easy then for them to return back up to EF2A area.
Right, so if we do not find or encounter more dead reef,
Hagfish and a
we'll
sponge.
likely ask them to go back then.
Yeah.
Dirty barrel. Look at that dirty barrel. Such a dirty barrel. Wow. It's impressive.
If we do encounter more there, then sky's the limit for them. They could do a transect on it, a little photomosaic, and just keep going south. One feature. Is it possible now to can you give me a ballpark distance between where we are now and EFG2?
Sure.
And then between EFG2 and EF2A?
To EFG2, it's 140 meters. And then EFG2A, not seeing that target.
To the north, it's just to the north where we were before at the
Oh, right up there.
West.
EF, okay, got it. Yeah, EF2A. Let's see that back. 300 meters.
I didn't get much sleep last night when I decided.
So Sam, I'm going to steal your attention. We're off screen and Rob nav here.
Oh, thanks. And we are about to end this move in 2 meters.
Okay, perfect.
And the transect, so the ship move is in 2 meters. We've just crossed the 200 meter mark with Herc, so I'm going to drop a target here. And the ship has 2 meters to settle out, so I think now's the time to look for zooms. What are we zooming on, shoreside?
Got it. So there were some remember I said that was an enormous or massive sponge. We passed by a couple. They weren't the large hetero